It's wonderful to see you all here. It's especially wonderful to see so many of you hanging around for this session, because if you want to know what stress feels like, stand between a crowd and whiskey tasting, all right? I will try to be short. What could I possibly tell you after a day of science like we've had that might be interesting or relevant in relation to this topic? Maybe nothing. All right, which is why I'm expecting some of you to go and stand in front of the microphone, and I will probably call people out if you don't stand up. So just be ready to get involved in some dialogue. Why do I say that? Because I've been thinking that you can't be inspired by things that you don't see. And over many years, many of you have inspired me greatly. So with all due respect, I'm going to try and repay that favor and do something here that might be useful. We as a society, I think, need to try and nominate tricky topics, sticky questions in the field, and bring them into an environment like this and discuss them. Not have a one-way dialogue, multi-directional, okay? So we're going to try and talk about stuff. This was the alternate slide I was thinking of using. Mistakes. It could be that the purpose of your life is only to serve as a warning to others. So if this works well, congratulate the executive and tell them you like this as a concept and you want to see more ideas tabled for discussion like this. If it tanks, it's all on me, all right? So that's fine. So moving right along then. The objectives here are really to just give you a very brief overview of what we've been thinking about as a society, or at least as an executive. For those of you who haven't gone to the Hoover site, to go and seek out the survey part of it, and then, time permitting, to get you actually having a discussion around some microphones about what an animal model consensus and the future of animal-based research might look like. So why are we thinking about all of this? Well, you're all very talented graphic artists, that much is true. But I think what we can say is this is a really complicated problem, overlaid on top of the diversity of people, both genetically, lived experience, comorbidities, everything else. And then in all of that, you're supposed to try and find causality. Yeah, good one. But pick an easy topic. Sometimes I think it would be easier to understand the biology of cancer than it would be the biology of cachexia. I put this part up here on the side because I think we often consider cachexia to be this sort of start here and there, but maybe there are multiple routes. Maybe it is multiple routes to a common end destination. And that's something that maybe we're going to find will matter when we start talking about precision or optimized, personalized therapies as well. Biomarkers that matter for one and not for another, and so on. So yes, you could study humans only, but how easily would you get to the end result of a successful outcome? In addition to that, why else might we need a consensus on animal models? Because we live in interesting times. I'm pretty sure you've all seen one version or another of these documents, whether it's the EU working on a mandate to try and eradicate animals from research and testing, or NIH and FDA discussion about changes that you no longer need animal models to be able to take something to a clinical trial. Now, it may be that you don't need an animal model for toxicity, but you might still need it for efficacy. I guess it's still on the FDA to decide. But these are discussions that are taking place, and unless we get involved in them, we'll be the recipient of other people's decisions. This one here is the Australian Science Agency weighing in on the matter as well. One thing that I thought was interesting is that in this whole document about positioning to move towards organoids as an alternative to animals, this color code shows you the organoids that they think are ready to be equivalent or superior to animals, as opposed to organoids that are maybe not likely to deliver the quality of results you need in the next 15 years, and those which they think is like no chance. And I think that when we consider our condition, we're talking about one where you've got to plug multiple organoids together. So some of them happen to live down here. So let's just take hard and then add that with hard and then make it impossible, you know? So therefore, in the face of this conversation about animals, are they relevant? Are they useful? Should we only be studying humans? Will organoids save the world? The society feels that there's a necessary document that needs to be developed, and we would like your involvement in developing that to make sure it's the best product we can make. Essentially, our position is one where we think that a smart approach to moving as far forward and as fast as we can involves using multiple models, which includes rodent models as well. Yes, maybe organoids might work, but ultimately we really need human on a chip with multiple organoids talking to each other. And I think it's probably worth mentioning that in something like Tabula sapiens, which was I think about half a dozen humans that were surveyed, 500,000 cells across 24 different tissues, more than 400 different cell types were molecularly characterized. So if you would like to make me a muscle organoid out of 2 cell types, that's great. I'd prefer if it had 13 or 14 or something like that. Same applies for the liver, same applies for the lung. And I think ideally, I'd like them all to be made out of cells that started in me so that we make sure it's my response, not your response or someone else's response. So there's a bit of work to do to be able to get that. I think, is there anyone here who works with organoids? Hand up? There's a couple, good. Maybe you can shoot me down later, but I think that to say down tools with the mice today is probably a bit premature. But I think we need to keep using them, but we need to use them better. So to a couple of quotes then. You may be familiar with quotes from George Box, such as all models are wrong, but some are useful. I think it's important to see the wider context of that statement. Specifically, all models are approximations. We actually heard that word earlier today. However, the approximate nature of the model must always be borne in mind. Or a better one perhaps, remember that all models are wrong. The practical question is, how wrong do they have to be to not be useful? Maybe that's the way we need to be approaching these sort of things. I thought maybe these would be better quotes, easier for me to, they resonate with me. In sport, you play to your strengths and you train your weaknesses. So maybe the science equivalent of that is we use models or approaches for their strengths, what they can give us, and we start to ramp down on the sort of activities that maybe are vulnerable to the weaknesses or limitations of those models. That's the sort of position that we as a society, I think, ought to advance. And that's the sort of discussion that the executive has been having so far. Essentially, we'd like to put forward a document which says that we don't believe that we'll advance as far or as fast without strategic use of rodent models, but acknowledge that like any model, they have their deficiencies. So there are both opportunities and limitations that we need to be mindful of with their implementation. So therefore, this starts to work towards some of the questions that we want to discuss. A document that identifies some of the advantages of using rodent models, some of the weaknesses or limitations we need to be more mindful of to avoid, discussion around aiming for best practice, and then aspiration for better practice, if you like. So very quickly then, I'm not going to go through here, that would be insulting your intelligence. There are a lot of things that we already are painfully aware about as far as differences between animals and humans. There will always be challenges as far as things like a survival study. There are humane considerations in animal models that limit us from really being able to study survival in the way we might perhaps want to or like to. But there are other tremendous things that we can do with rodent models that you just couldn't do in a human setting, and I think we should not forget that or lose sight of that. So in terms of starting with trying to identify or define best practice, again, many of you will be aware of things like the ARRIVE guidelines. These are supposed to be the minimal sort of reporting elements for a document. What we would like to do is perhaps build on that with a set of guidelines which are more specific to cancer cachexia related research. Here are a number of elements or attributes, if you like, which are in the Hoover survey as a starting point. Again, it's not comprehensive, it's a start, a catalyst, if you will, to sort of knock loose better ideas from some of you out there. So we want to hear what you think about these. We also want to hear what you think about additional elements that ought to be reported and factored into your design. And then aspiring for better practice. So if you were to gaze into a crystal ball, such as this one that you may be able to find somewhere around town, what does the future look like? Is it possible that our landscape may be completely inverted in a few years because of things like organoids? Or is that actually further off in the horizon? So the discussion around better practice starts with, for instance, always loved these graphs, Vicky, they're beautiful. Do we need different models that represent the different types of cancers? Do you, for instance, embrace the diversity of phenotype within the genetic diversity of mouse models. So this is the mouse hybrid diversity panel, showing you the variance in lean mass across the different strains. If you focus on a black 6 mouse and a Balb C, maybe you're not really using the model that's the best model, most representative. So this is something we should be thinking about too. Is it possible that we're looking long-term at multi-site collaborative studies where you validate your observation with other teams? This particular paper that some of you may have seen recently enrolled 2,500 mice, I think it was, or 2,500 rodents across multiple labs to study stroke medications. Or would you perhaps feel that better practice might even involve an equivalent of some sort of consulting board that you can go to for advice in how you want to do your design? Something like a CTAB, which is almost a segue to our next speaker. So again, these are some of the things that we've thought would be good to talk about, and we really would like to hear what you think. We'd especially like to hear what we've missed, what else you think is worth contributing. So just to point you there, in surveys, you'll find a specific survey about animal models. These are some of the questions that we have posed to you, specifically what you think should exist in best practice, what you think we ought to be aiming towards in better practice. What do you think are the goals and expectations that we should be putting on ourselves? What are the challenges that we should be trying to meet and solve in moving forward together? And hold ourselves to those sort of standards going forward. And are there other questions that we should be asking? I'll bring that up in a minute. I just wanted to acknowledge these people because they have been the ones that we've been bouncing ideas amongst so far. But we think that, again, we're starting and we're encouraging you to opt into that as well. So that's going to go pretty slow. But what I hope we'll see as these T-shirts come to shape every couple of years is that we'll see emergence of additional words representative of new models, new approaches, some of our consensus position being developed as well. So I've put these questions up here again. I'm going to stop now and encourage some people to stand in front of the microphone. You don't have to answer them in any order, but I would love to hear what you think about some of these ideas. So thank you for your attention. Anyone brave enough to have a first go at offering an opinion?
Hey, Paul. One thing that I completely agree, and in general, I think the mice I used with the LLC and the C26 that are subcutaneously implanted are so far away from the truth. So I do agree. This is a very good discussion. The only thing that might worry me in relation to for example, some of the genetic models where you can get spontaneous tumors development and so forth, so more of what you see in a human or in a patient, is that it takes so much longer. And we compared to 30 years ago, where you can make a very nice paper and high impact and all these things with a very few amount, a couple of data, a couple of figures, and it's a perfect, nice story. Now you see, if you want to aim for higher things, then you need 5 primary figures with 10 panels and 10 supplementary figures. And I mean, and if you want to do so many things, you only have so much time as a PhD or a postdoc. You don't have 10 years to do it. So you also need a model that is somewhat reliable, fast. So I don't know how to, I still think we should aim for the better model to actually get closer to the truth.
Agreed.
So I agree with that, but it comes at a cost for maybe the researchers that doesn't have tenure.
I think it comes at a cost for everyone. So again, this is another one of these questions or elements that we as a field have to weigh up and balance and say, how important is speed versus clinically meaningful, accurate model? I think that's part of the allure that is advertised with things like organoid systems or body on a chip, is that you might be able to move through things faster. I would love it if someone could take cells from me and create a human on a chip, and then we could study it and see what happens. But I don't think we've got it just yet.
Hi, thank you for that talk. I do agree that cost can be an issue for some of these longer-term models, particularly for smaller labs trying to enter into this field. But I do think that a lot of things can be done just by appropriate data representation within papers. We just published a paper where we did a data extraction of every single C26 paper published from 1990 to now, and the way that data is presented and what data is presented and the way it's presented is vastly different. And so we, in our own mice, did that same analyses and found statistical differences between like using actual weight versus percentage of body weight versus percentage of tumor-free body weight. And so some just basic like standards across the field for how we should be presenting data in papers, I think could help us a long way with like translatability and comparison across studies.
Agree, again, 100%. So I hope I'm not coming off as preaching here. That is the last thing I wanted to do. I'm consulting on behalf of our executive. What we're trying to do is encourage people to get involved. It's an opt-in activity here. We want people with good ideas to help us define what we should hold ourselves to as far as the design and the reporting and so on. So it's a great comment, and we love you for putting together that paper comparing all of the different C26 studies as well. I would like to ask some, oh, please.
I really like the idea of trying to have a data repository, one for anyone who already is obviously still doing Cachexia research, but anyone who wants to get into it. And then also some universities can't get access to all the papers or countries or whatnot. So it would be helpful if there was one place or someone they could go to get that. And then I didn't know for better practice if it would be good to include something about the animal facility itself, because I think people have done similar studies at different locations and they end up different. And it might be personnel, but it might also be the facility.
Those are the sort of things that can keep you awake at night, wondering about what exactly is driving that phenotype that you've got. So I agree. Look, I'd say personally, some of the most satisfying research collaborations I've had have been where things we've done have checked out when collaborators have used them themselves in their own lab. I think that we will be better for those kind of experiences and interactions and the sharing of data as well. So I'm keen to hear if there's anyone here in the industry space who wants to tell us what they, as an industry person, think about the future of research. Do they think organoids are going to save the world? Is it where the company wants to go? Does anyone want to field that question?
I am not from industry, but from academia. What question? I didn't hear the word AI and machine learning. Why, as a field, we are not getting there? Because probably part of the solution and the help AI can give us really be a tool that we can connect the data. In addition, what you suggested, I don't think they're all mutually exclusive. We have to replace one with the other. Organoid will never replace animal. Animal will help us address certain questions, but will not probably tell us what's the drug that will solve the issue. We need human setup, and organoid can offer that. So it's really, I don't see that we can push that. We need to exclude one of the tools that we have. We need to find a way to really, certain type of question can be addressed only with even in vitro cell culture, like mechanistic details. We can test them in organoid, which will give us a better setup than probably and easier testing different steps than animal, yep, etc.
I agree. To me, standing here, it looks obvious that you would use all the tools at your disposal. You wouldn't leave certain ones off the table. The problem, of course, is that animals have a lot of emotive baggage around them, right? So there are definitely forces at work to try and remove them from the arsenal. I think it's on us to explain why sensible, humane, rational use, when appropriate, is still going to be necessary. I guess that's how I'd put it.
Yeah, and using AI can help us build model that can, like now many area like drug screening uses AI like.
I agree.
And as a powerful tool, why we can think about how to include it.
Yep. So, I mean, you will see talks over the remainder of this meeting about how AI might help, which is great. And maybe that's another topic that we will table and try and get people to discuss in the future as well. So, look, I think I've pretty much run out of time, and you have a more interesting talk to hear, after which I believe there's a session on how to be competitive with grants. Is that right? Or is it whiskey drinking as opposed to... Anyway, thank you.